Okay, welcome everybody. I think the room is already full, so we can start a couple of minutes early. Thank you and welcome to this session about the role of mitochondria in autoimmunity. It's a subject that's personally really changed my thinking over the last few years as somebody who was brought up to just think about apoptosis and genomic DNA. And we have three superb speakers for you today. So the first of those is Virginia Pasquale. Virginia works in New York and has made a number of significant discoveries in the pathogenesis of lupus, particularly on innate immunity that are too numerous for me to list. So I'm really looking forward to her talk today. Over to you.
Thank you very much, Ed, and thanks a lot to the organizers and to Sana and Ed for chairing this session. It is a real pleasure to be here. I have a fundamental disclosure for you, and it's that I did not choose the title of my talk, so I have slightly modified it. I would never dare to talk about the origin of the response to DNA lupus, especially in 20 minutes, and I am not even qualified to discuss that topic. So what I'm going to do instead is I'm going to give you a little overview of the fascinating world of mitochondrial DNA as a driver of immune dysregulation. I will review some basic concepts about it, and then I will show you, or will remind you of some of the data that my own group has published through the years, and move from my own data to some more recent data that will eventually be also discussed in this session, and I think brings the topic to a very, very high level of interest for us rheumatologists. These are my real disclosures, although I have no conflict whatsoever with this talk. So when we talk about responses to DNA in lupus, obviously we all think about nuclear DNA. And this comes from the seminal discovery of Dr. Hargreaves back in 1948, when he described as the truly first biomarker of lupus the presence or he identified the ELI phenomenon. So the capacity of lupus serum to modify the nucleus of cells, induce their opsonization into mainly phagocytic cells that would, this nuclei would occupy basically the cytosol, displace the nucleus of the recipient cell. And that was a phenomenon, although not totally specific to lupus, really very, very, very associated to the pathogenesis of the disease. About 9 years later, Dr. Henry Kunkel, whom you all know, working at the Rockefeller University, identified that factor responsible for the ELIF phenomenon as antibodies, IgGs, binding to DNA. At the time, it was thought this is mainly, almost exclusively nuclear DNA. He showed in this Science publication in 1957 how those purified factors, those purified gamma globulins, could really induce the visualization of this material that it was binding in the form of nuclear DNA. So the world, our world, the world of rheumatology, has been using the measurement, the quantification of anti-double-stranded DNA for decades as one of the criteria for lupus diagnosis. We know that these antibodies are very specific, and we really talk about nuclear DNA, nucleosomes, etc., as the main form of antigenic DNA. However, well, where does this capacity to develop anti-DNA antibodies comes from? The work of Michel Nussensweig and many others in the field have been telling us in mice and humans that we all have the capacity to develop basically every autoantibody specificity. And with Hedda Wardeman, Michel showed back in 2003, sequencing single B cells and cloning and expressing those antibodies, that the majority of the bone marrow repertoire of pre-B cells is autoreactive, but there are checkpoints that control the expansion of these autoreactive clones, and both in the bone marrow and in the periphery, there is a selection such that this 75% of autoreactivity comes down to 5 to 20% of autoreactive B cells coming out of the bone marrow as transitional, eventually naive B cells. A couple of years later, with Sergei Jurasov, and actually I was part of this study and everything that was clone sequenced, expressed here, was coming from our clinic, pediatric rheumatology clinic, children with lupus in Dallas. Sergey and Michelle showed that in kids with lupus, this peripheral purging fails. And in these patients, the naive B-cell pool reaches the periphery with basically 50% of them carrying autoreactive receptors. So we know that. So every one of us has the capacity to develop anti-DNA, anti-RNA, polyspecific, monospecific autoantibodies. But normally, we keep them pretty well under control. And we know that if we screen every one of us today, probably none of us will have detection of anti-double-stranded DNA antibodies. So what makes DNA immunostimulatory? There is more and more information about different forms of DNA, and a few principles come over and over again. Is DNA normally associated with proteins, anywhere from histones to antimicrobial peptides to other kinds of bigger molecules, bigger proteins that have the capacity through their mainly charge to bind DNA and then being delivered through specific receptors or through endocytosis to cells where TLR9 can be activated. We also know that modifications or special forms of DNA, like those that are CpG enriched or those that are hypomethylated, are better ligands for TLR9. Definitely modifications, especially in the form of oxidation, induce a resistance to degradation, and in addition, will enhance ligation not to TLR8, but to TLR9 and also to cytosolic receptors, as we will see later. The cytosolic localization, where they can easily access cytosolic sensors like cGAS, mitochondrial origin, that is CpG-rich, hypomethylated, and as we will review, bears some very, very peculiar characteristics, expression in extracellular compartments, for example, through the extrusion of nets. And obviously, if we have a lot of DNA that is not being degraded inside or outside the cell, this is going to saturate the sensors and eventually make those nucleic acids more immunostimulatory. It turns out that basically every one of these characteristics is shared by mitochondrial DNA. Mitochondrial DNA is a has a fascinating structure and biology. It's a double-stranded DNA, circular DNA, measuring 13 kilodaltons. The replication starts at this light strand promoter, and then it creates either the full-length chain of the mitochondria, strand of the mitochondria, but it also stops frequently, generating extra strands of DNA that can align to the double strand and create these very peculiar D-loops that are stable three-stranded structures that, as you can imagine, are going to be more immunostimulatory. They can bind sensors like cGAS with higher affinity than just plain double-stranded DNA. Obviously, double-stranded DNA, especially when it's modified, is a very good ligand for TLR9. We already reviewed that. And when it comes to transcription, the mitochondrial DNA has a very peculiar characteristic, and is that it's being transcribed from both ends, through the light-stranded promoter in one direction, through the heavy-strand promoter in the other direction, generating a double-stranded RNA structure that is also modified, uncapped, and can be a pretty good ligand for cytosolic sensors like RIG-I of NDA5, and then eventually lead to the oligomerization of the mitochondrial surface molecule maps. So we can see here how because of these peculiar characteristics of the DNA and its transcriptional downstream forms, it becomes a potentially pretty immunostimulatory form of nucleic acid. Now, mitochondrial DNA is not naked for the most part. It is, like chromatin DNA, very tightly bound to cationic proteins, in this case, to the transcription factor A of mitochondria, or TFAM, which is a very interesting a structure that has more or less this shape, and when it binds to mitochondrial DNA, it's going to induce the bending of the mitochondrial DNA, making it compact into what is called nucleoids. Nucleoids will be the mitochondrial equivalent of the nucleosomes coming from nuclear DNA. This is important, as you will see later in the next slides that I will show you, but keep it in mind. So mitochondrial DNA is not naked. Normally, because of its amazing immunostimulatory capacity, mitochondrial DNA is going to be enclosed within mitochondria. And when there is mitochondrial damage, in this very nice review by Jerry Shadell, we can see what I was telling you earlier, that the mitochondrial DNA is shaped around this bended transcription factor TFAM because of the potential immunostimulatory capacity of this DNA is enclosed within the double membrane of the mitochondria. When there is mitochondrial stress or mitochondrial damage, mitophagy is going to take care of it. The mitochondria with the DNA and the RNA is going to be degraded, and the immune system, for the most part, is not going to see these nucleic acids. However, there are so many instances where mitochondrial DNA together with TFAM enclosed by double membranes can leave the cell, where this can also be internalized within cells. It can reach cytosolic compartments like the lysosome, where it would be degraded eventually by DNase II. But if there is an imbalance between the amount of degradation and the presence of this DNA, especially if it is modified and less sensitive to the degradation, it can reach the endosome, where it's going to activate TLR9 and give rise to production eventually of interferon. Mitochondrial DNA naked, together with TFAM, can also leave the mitochondria and reach the cytosol, where it can engage cGAS and STING and also lead to production of interferon. And then mitochondrial DNA is not always intact around TFAM, especially when it's oxidized or when the mitochondria is replicating itself. We are going to find short stretches of DNA that eventually can also lead the organelle and reach the cytosol, where it can engage a completely different set of receptors, including AIM2 and the inflammasome, like NLRP3, leading to the cleavage of caspase, cleavage of pro-IL-1 beta into mature IL-1 beta and IL-18, giving rise to a very pro-inflammatory phenotype. And now these cytokines can be released in different forms. So we can see here in this snapshot all the potential sensors for mitochondrial DNA, and we are not even covering the sensing of mitochondrial RNA, which will be another a topic for another day. So with that in mind, we in Dallas originally and now in New York have been trying to understand the signals that lead to activation of different sensors and immune activation in children with lupus, knowing that if we do immunophenotypic follow-up of these patients, we can capture signals from interferon, from myeloid inflammation, from B-cell and plasma cell activation. So we have been engaged in this exercise for many years. And through these exercises, we have come to the realization that there are immune cells, like neutrophils, and actually red blood cells, whose mitochondria can be immunostimulatory. And I'm going to summarize this very briefly for you. Our main messages are, we found with Simone Caielli, who is my collaborator for many years, that lupus neutrophils, under certain conditions, extrude mitochondrial pieces that carry oxidized mitochondrial DNA. We have also more recently found that lupus red blood cells leaving the bone marrow have not totally degraded the mitochondria, they retain it, and these mitochondria can eventually become very immunostimulatory. So let's review this very quickly. First of all, we know that neutrophils, through the work of Arturo Cislinski and many other people, including some of you in the audience, neutrophils under certain forms of activation are a spectacular source of extracellular nucleic acids. They can depolymerize the nuclei and send nuclear DNA to the extracellular space bound to proteins, histones, and even cytosolic proteins that can definitely activate the immune system. This is supposed to be a mechanism of defense to trap extracellular bacteria and fungi, but sometimes, in patients with lupus in particular, can become really a upstream event in immune activation. Now, before, or a few years after Arturo described NETosis, the group of Dr. Simon in Lausanne, in Switzerland, described that actually neutrophils don't need to depolymerize the nuclei and die in order to release DNA. But in the steady state, human neutrophils release pieces of mitochondria that contain DNA and may or may not be bound to nuclear DNA. That was a seminal paper that really passed a little bit under the radar compared to the spectacular publications from Arturo. So a few years after we saw the work of Dr. Simon, we were looking into this phenomenon, and we confirmed that when we put healthy neutrophils in vitro, a few minutes to hours later, we start recovering from what looks like viable neutrophils. We start recovering pieces of mitochondria that contain TFAM-bound mitochondrial DNA. When we expose this DNA to plasmacytoid dendritic cells that are the main cells in the immune system, together with B cells that express endosomal TLR9, we saw very little activation. However, when we recapitulate a lupus environment, so we prime with interferon that activates or increases the expression of TLR7, for example, and we expose these cells to immune complexes that contain RNA, anti-RNP antibodies, we see that we confirm the extrusion of these mitochondrial pieces with TFAN bound to DNA, but now the DNA is oxidized, and when we expose PDCs to it, it induces very, very significant amounts of interferon production. So that led to a publication, and Christian Lod published very similar data the same year in Nature Medicine. We went to try to understand why this was happening, and I'm going to go very quickly through this. The bottom line is, in basic human neutrophil biology, when the mitochondria of neutrophils gets damaged, it's not removed by mitophagy. Rather, what happens is, especially in the presence of oxidation of the mitochondria, that pieces of oxidized mitochondrial DNA that you see here in green leave the mitochondria towards a lysosomal compartment where they get degraded. This doesn't happen in monocytes exposed to oxidation. It's a very peculiar feature of the neutrophils. So then what we saw is that these pieces of oxidized DNA go into a vesicular compartment described before by Heidi McBride, and they are fused to some mitochondrial proteins to show that this oxidized DNA is definitely of mitochondrial origin. What happens next is, in healthy neutrophils, we see these green dots of oxidized mitochondrial DNA. In a lupus recapitulating environment, we see more of these green dots, and we see aggregation of these green dots. And the main difference comes when we co-stained with mitochondrial proteins. Here we see the external membrane of the mitochondria stained with TON20. You see that TON20 in red is not colocalizing with oxidized mitochondrial DNA in green, because this DNA is leaving the mitochondria very efficiently. In lupus conditions, these green dots are not living so efficiently, and they get stuck to TON20. That means they are retained within mitochondria. If we take the blood of healthy children or the blood of children with juvenile dermatomyositis, we recapitulate this in vitro phenomenon. The green dots are leaving the red membranes. In children with lupus, the green dots staying in the mitochondria. You see this yellow. And now when it's extruded, we are finding outside the cell pieces of mitochondria with oxidized mitochondrial DNA. So that was very interesting to us. We look further down. We expose this oxidized TFAM mitochondrial DNA to PDCs. saw the presence of interferon and then exposed naive CD4 T cells to it. And in this paper that we published in Nature Medicine in 2019, we identified the adaptive immunity consequence of PDCs being activated by this material. And what we observed was the generation of a peculiar phenotype of helper Th1-like cells producing or making interferon gamma and IL-10 that, to our surprise, were not regulatory based on the production of IL-10, but they were very good B-cell helpers. So I'm not going to go more into this. This was published years ago. We re-identified these cells last year through single-cell RNA sequencing. The important feature here is that starting with a mitochondrial oxidized DNA coming from an innate immune cell, we are shaping the adaptive immune response into a pro-helper phenotype. So that's all downstream of the mitochondria. Of course, neutrophils are not the only cells in the immune system that release mitochondrial DNA. Eric Boilard, who will speak after me, has done beautiful work showing that platelets do very similar things. There is precedent with eosinophils, with basophils, and many other or several other forms of immune cells that can extrude these immunostimulatory forms of DNA. And very recently, and this is a paper in bioarchives by the group of Ryan Flynn at Boston Children's, showing that it's not only that these pieces of DNA, mitochondrial DNA, are released into the extracellular space, is that they can also remain bound to the surface of cells. In this case, these are B cells. The white that you see here is oxidized mitochondrial DNA. Ryan doesn't know if from the cell intrinsic extrusion and then religation or coming from other cells, but it sticks to the surface of B cells and is only removed in the presence of DNase 1-like 3, the main restriction factor for recognition of nucleosomal DNA. So you see here another peculiarity of this amazing mitochondrial oxidized DNA. DNase 1 does not degrade efficiently this material. It has to be DNase 1-like 3, and Ryan has shown, or is showing in this paper in review, that patients with DNase 1-like 3 missense variants have an increased level of surface DNA, mitochondrial oxidized immunostimulatory DNA on B and T cells. I think that in these 20 minutes, I'm not going to be able to show you much more. Just to tell you that, of course, Eric and Paul Fortin have recently published a very, very seminal paper that obviously involves the amazing collaboration of many groups around the world, recruiting many patients with lupus and looking for the presence of anti-DNA antibodies that are not just anti-nuclear or nucleosomal antibodies, but antibodies that bind to intact mitochondria, and there are many proteins and lipids that accompany it, intact mitochondrial DNA, and also, again, we didn't touch it, but phenomenal chapter in immunostimulation, mitochondrial RNA. So these are important because actually their presence is associated and predicts nephritis, arterial vascular events, mortality. So I think that the world of recognition of DNA is now switching, is not new. Back in 1984, so many, many years ago, the group of Dr. Tan already showed that there are antibodies recognizing native DNA that bind to mitochondria. So this is a long story that we have been overlooking, and I think it's about time for it. I'm going to pass this section. I was going to show you that in red blood cells, mitochondria also induces not only interferon, mitochondrial DNA from the remnants of red blood cells, induce the production of not only interferon, but also IL-1 beta, a phenotype that is extremely pro-inflammatory and that we can actually identify in the glomeruli of children with proliferative lupus nephritis. You see here one glomeruli with cells that stain for MX1, an interferon-stimulated protein, as well as interleukin-1 beta, and we have published, and other people have published, that these cells can be extremely immunostimulatory. So the story is only beginning, This is the summary. Cell-associated and cell-free mitochondrial DNA, and let's not forget RNA, are phenomenal sources of immunostimulatory nucleic acids. Oxidized mitochondrial DNA degradation is altered in lupus neutrophils and is a phenomenal interferon inducer. Oxidized mitochondrial DNA, both the full genome and short replicative forks, can be retained within lupus red blood cells and activate when they leak into the cytosol of the opsonizing myeloid cells, activate cytosolic sensors, and lead to interferon and IL-1 beta production, and we are following these cells now through the tissues. Lack of proper degradation of mitochondrial DNA not only induces activation of the innate immune system, I show you, is also upstream of what I think are very important adaptive immune alterations in lupus, and as others will show you, this is not our work, a variety of mitochondrial antigens are truly recognized by lupus autoantibodies and can be phenomenal biomarkers of lupus severity, activity, and even mortality. So for the part from my lab, I have to thank extensively Simone Caielli, who has been a driving force in everything we have published. Prita is a very talented postdoc working with us. Jingwa and Uthra, bioinformaticians. Roman and Sun-Hee gave us years ago the initial hints that neutrophils and red blood cells could be involved. The most important part of the slide is the clinicians in Dallas and New York who follow the patients whom we studied, and as always, the patients and their families. Thank you very much.
Thank you for this brilliant oversight. Do we have any questions? Yes, please.
Yes, thank you very much. I'm Professor Jacobsen from Copenhagen, and great presentation. And I think it was interesting to see the interaction between type 1 interferon and the mitochondria. So I was thinking antiviral mechanisms, shutting down energy metabolism in an infected viral cell is one of the mechanisms that type 1 interferons do. So I was just thinking, is interferon, type 1 interferons, are they sort of driving an oxidative crisis and then making antigens from the mitochondria more available? Or is it the antigen source from the mitochondria which is driving the interferon signature?
Well, definitely the second is true. There is no doubt that leaked mitochondrial DNA and RNA are phenomenal inducers of interferon. There is no doubt about that. For the role of interferon in shaping the mitochondria in a way that makes it more antigenic, I don't think there is a lot of data. What we do know is if we prime healthy neutrophils with interferon, this is a sine qua non factor. For then the anti-RMP or any other immune complex that is going to activate TLRs in the neutrophil for making that process of, and I didn't go much into it, decoupling the oxidized DNA from the TFAM transcription factor. That is a fundamental step in routing the oxidized DNA to the lysosome under healthy circumstances. So definitely interferon plays a role there in avoiding the decoupling from TFAM and therefore making this routing not possible. So that we know. Now, whether interferon or interferon-inducible molecules, and I'm sure there is literature, so forgive me for my ignorance, whether that can shape modifications of mitochondrial antigens, that is possible, but I don't know that well, that literature. Definitely the metabolic aspect of it that you mentioned is fundamental, and I didn't even go into that, or into the connection with lipids and many other aspects of mitochondria that definitely I didn't have time to cover.
Okay, I think we can just manage one more question briefly over there. And
great talk, thank you.
It
will be brief. Great talk. Do you see the oxidized mitochondrial release from all lupus neutrophils or specific subsets?
I couldn't hear.
Do you see this oxidized mitochondrial release from all lupus neutrophils or specific subsets? Do you see it in normal
Oh,
density?
yes.
And
Yeah, in our own studies, we only used mature neutrophils. Obviously, we have described many years ago that low-density neutrophils and early neutrophils were expanded in lupus. That was a 2003 paper. Mariana Kaplan obviously has done fundamental work on the low-density neutrophils. So Mariana, I am not sure. Christian is here. Maybe he can comment. We mainly looked at mature neutrophils. We never looked at the capacity of earlier forms or even low density. But I'm sure that Mariana and Christian have looked at that. So he can tell us later, maybe.
Okay, we can discuss it maybe more at the end. Thank you very much. But we should turn to our second speaker. So thank you very much. So Dr. Eric Boyard is a professor at the Department of Infectious Diseases and Immunity at the University of Laval, and he's published seminal discoveries on the role of platelets in autoimmunity and in lupus and their microparticles and mitochondrial components. I will leave you to say it better.
Thank you. Thank you very much for the introduction. And I wish to thank the organizers, EULAR, the chairs, for the invitations. I'm very grateful for this. I also didn't choose the title, but I thought I will use it. It reads like an answer, and it will come as a question, you see. Yeah, those are the COIs. So you've traveled to come here, and we all travel, commute somehow. And in the presentation, you see that not only us can travel, mitochondria can travel as well. They can travel between cells, they can travel between tissues. And so in the presentation, I'll talk about transfer of mitochondria between cells and potential clinical application. We see that platelets can be a source of mitochondria. And we talk about this context of platelets release of mitochondria in inflammation, and then allude to adaptive immunity. So there's a rapid and fast-growing literature on evidence of mitochondria being released from cells. I thought that, of course, I will not review all of that literature, but I use that timetable to illustrate some of the findings that were published. It's in the late 70s that it was found that yeast that were deprived of mitochondria could be cultured with mitochondria from platelets, and these mitochondria will be captured by yeast, and yeast will use these mitochondria. So that was the first example of transplantation of mitochondria in plants. Then it's been described mitochondria from one flower can travel to another flower, even from a different species, through pollen and bees. And then more and more studies show that mitochondria could travel between cells in brain, heart, in cancer, but also in sepsis, inflammation. It's in 2014 that we've shown that platelets could release their mitochondria, that there were some levels of mitochondria in blood in healthy conditions, but that could be increased during inflammation. And now there is more and more studies that aim to actually use this finding and apply them to clinical applications, such as in this one where the investigators work with young kids that had a lethal disease due to mutations in their mitochondrial DNA. In that case, they isolated the CD34 cells from these patients, which they incubated with mitochondria from a healthy individual, transformed or transplanted their mitochondria in the CD34 cells, re-injected them in patients, and the patients showed improvement for the first 6 to 12 months. There are evidence in mice where the investigators injected healthy mitochondria in mice with Lee syndrome and Parkinson's, which are a disease in which mitochondria have defects, and they could improve the disease. And now the attempts to improve the efficacy of transplantation. In this case, the authors have designed or modified the surface of mitochondria so that they can be targeted to specific cells or tissues. And more recently, which last month, in fact, it was found that mitochondria, which they isolated the mitochondria, and if they are encapsulated into the membrane of red blood cells, these mitochondria were more efficient at being captured by other cells. And that study the authors could find that they could protect mice from Parkinson and Lee syndrome as well. So these are evidence of or suggestion that we may leverage findings made on transfer of or travel of mitochondria to clinical purpose. And right now, there's a lot of studies that aim to determine what would be the best source of mitochondria so that we can work with humans, what would be the best approach or best targets with this mitochondria and transplantation. So that the mitochondria are released, there must be some principle of release, of course, and there are these three main mechanisms that have been described. One which is called passive, which implicates cell damage or release of debris, including mitochondria. In this case, DAMPs or molecules from mitochondria can activate different components from the immune system. And of course, stimulating the immune system can be good. It can promote wound healing, for example, but if this is chronic, then it can make, it may contribute to inflammation. There's a release of mitochondria through membrane vesicles, so that could be mitochondria wrap into extracellular vesicles. It could be component of mitochondria within vesicles as well, or actual naked mitochondria that can be released from some cells. And the transfer of mitochondria can also involve tunneling nanotubes. In this case, these tubes form between cells, and components from the cytosol can travel between cells. So in our lab, we're interested in platelets and their role in immunity. You may know that platelets are not exactly cells. They're small fragments from a larger cell called the megakaryocytes, which is a cell that we find in the bone marrow, in the lungs as well. And these cells, they are unique. They can replicate their DNA several times without cell division. So they can replicate DNA up to 128 times, which is amazing. And so that the cytosol is very rich in molecules, including mRNA, microRNA, cytokines, and mitochondria. These cells then will form these membrane protrusions, which will protrude into blood sinusoids and will break down with the blood share into smaller platelets. So one megakaryocyte can give rise to about 5,000 platelets. And these platelets in blood, will, as you can expect, they are well known for preventing bleedings, but they can also contribute to immunity. So we counted mitochondria in platelets. We found an average of 4. So sometimes we see 7, sometimes 1, never 0. So an average of 4 mitochondria per platelet. And considering that megakaryocyte can make up to 5,000 platelets, you can imagine that megs can have 20,000 mitochondria, which is huge. Cells that are very active, such as neurons or muscles, will have 2,000 mitochondria. So it's a lot of mitochondria for a cell that we don't expect to be using so much energy. So in our studies, and it's published, so I don't want to spend too much time on this, but we found that in lupus, and by working with patients and mouse models, that platelets could release their mitochondria. Some of them were naked, some of them were within vesicles, and this is mediated by the activation of platelets by immune complexes that will activate Fc gamma R2, which is the only receptor for Fc receptor for receptor for immune complexes expressed by platelets. It's been replicated in different diseases, including RA, and we found that the transfer of mitochondria to other cells could also transfer the potential of bioenergetics of platelets to, in this case, macrophages and neutrophils. So then it'd be great if we could validate whether this transfer or release of mitochondria is promoting disease. Would that promote lupus? And the experiment that we would like to do will be to inject mitochondria into volunteers, a lot of volunteers, inject mitochondria that would be intact, mitochondria that would be damaged or oxidized. Now, I don't know how's your ethical human board, but it was quite complex to even dream of. And but we can get around this by thinking of platelet transfusion, at least partially. Platelet transfusion is a very common medical procedure. 50,000 platelet transfusions are taking place on Earth every day, and 1% of them can induce inflammation, can induce adverse reaction. Sometimes more rare, can induce death, literally, by inducing inflammation into the lungs. So we thought that perhaps platelets, when they are isolated and banked by blood bank and stored in platelet bags, perhaps they release their mitochondria as well. And if they are transfused, these released mitochondria could perhaps promote inflammation in the recipients. So we decided to measure mitochondrial DNA in platelet storage bags. For this, we've quantified mitochondrial DNA in 70 bags that did not induce adverse reaction and 70 bags that did induce adverse reactions in patients. And we found that there was a significant increase when platelet concentrates induce inflammatory reactions in the recipients. And that DNA was not free-floating. We used flow cytometry to quantify vesicles, and it correlated strongly with vesicles containing mitochondria with centrifugation, proving that the DNA was part of particles. We could deplete them using CD41 depleting antibodies, showing that it was part of vesicles from platelets, and DNA was resisting to nucleases. So this was the work of our grad student Geneviève Marcoux in the lab. So this suggests that the platelet, the mitochondrial DNA derived from platelets may be a good marker to identify bags that should not be used for transfusion, or perhaps even less in individuals that are more vulnerable, like newborns, for example, or those with trauma. But it suggests that DNA or mitochondrial DNA may have an actual role in inflammation in humans. And in terms of mechanistics, there are some studies that were performed in mice. It was found that mitochondria from platelets could activate ROS in neutrophils. In a more recent study, it was found that extracellular mitochondria could activate neutrophils. It will prime them, I should say. And if combined with antibodies against HLA molecules on neutrophil, that will promote netosis, entraly in mice, and death. So if we look at it, like if I make this short summary, mitochondria are good. If they are released from cells, they can travel between cells, can be captured by a recipient, and there's even clinical applications that are being developed. Simultaneously, a different literature is demonstrating that mitochondria outside cells can be pro-inflammatory, may generate adverse reactions. And in lupus, we find mitochondria outside cells. So what does it mean? Is this a good thing or a bad thing that mitochondria are being released from cells? So to get more insight from this, we decided to consider adaptive immunity, thinking that, like it was well presented by Virginia just before, perhaps exposure to mitochondria in a chronic way perhaps will lead to generation of an adaptive immune response and breakdown of the immune tolerance and generation of antibodies against mitochondria. So this was the work of Jan Becker, a grad student in the lab, who developed these assays where we aim to measure anti-mitochondrial antibodies. And for this, we use mitochondria isolate from mouse liver, from a human hepatocyte. We isolated mitochondria, not using kit, using protocols that were very improved so that there's no Golgi, ER, nuclear components, and the mitochondria were coated in plates, were also coated mitochondrial DNA and mitochondrial RNA, and measure the antibodies. And we found that there were significant levels of antibodies against the wall or the surface of mitochondria, the anti-wall mitochondrial antibodies, anti-mitochondrial DNA and RNA in these patients. PBC is a primary biliary cirrhosis, an autoimmune disease in which there are anti-mitochondrial antibodies. They are negative in this assay. We believe this is due to the fact that in PBC, the targets of the antibodies are on the inner membrane of mitochondria. In our assay, we are measuring antibodies on the surface of mitochondria. So the big question was, would these antibodies be present at the beginning of the disease? Do they correlate with any disease manifestation? So for this, we were fortunate to work with a standing group of rheumatologists, the SLIC group, and with the work of patients as well. So we had these 1,100 patients with 3 different visits from year 1 to 7. and we had 130 healthy individuals as well, and we've measured these 3 types of anti-mitochondrial antibodies in these individuals. And we found the first thing that we can conclude is that the patients had anti-mitochondrial antibodies, anti-wall mitochondria, DNA and RNA from the start of the disease, and they remain significantly more elevated than in healthy. This is a dash bar. And you can see that mitochondrial DNA antibodies, they tend to go down with time, which fits perhaps with the treatment that is followed, while the mitochondrial RNA and anti-wall mitochondrial DNA antibody are remaining elevated. What's important is that we found that the anti-wall mitochondrial antibodies predicted mortality if they were measured within the first 3 years of diagnostic. The anti-mitochondrial DNA and RNA predicted nephritis. And another intriguing finding is that for the anti-mitochondrial RNA, these antibodies predicted some vascular events. In females, that predicted a poor prognostic, while in males, that was protective. So there's some link due to some sex differences in terms of anti-mitochondrial RNA. So this suggests that these antibodies can be used as a biomarker and maybe even more potently be used as a predictor. It would be interesting to combine them with other antibodies that are more traditional in the field. Of course, using whole mitochondria in an assay is not very convenient, as you can imagine. So we aim to determine what's the target of the antibodies on the upper or the outer membrane of mitochondria. So we isolated mitochondria and did experiments in which we isolated IgG that were bound to mitochondria and used mass spectrometry to identify them. And we identified mitofusin-1 as a main component, main target of these antibodies. So this is a protein involved in the fusion between mitochondria. We found that these antibodies were more elevated in patients with lupus. They were absent in PBC and APS. So now we have developed this. We aim to apply this to more high throughput approaches. It was conjugated with nanoparticles so that it can be, for example, robotized. So in this case, it was used with the Aptiva system from Werfen, and we've measured the anti-mitofusin-1 antibodies across 1,000 individuals here. And what we can see, is that lupus is obviously the most striking in terms of prevalence of anti-mitofusin-1. Although there's some spreading here with RA, which may be due to some non-seronegative and erosive patients, which was demonstrated by the laboratory of Christian Rood. And so it seems that anti-mitofusin-1 may be an additional marker of lupus. But how does it connect to the disease or the pathogenesis? Of course, there's a lot of work to do to understand this. But if we think that in normal conditions, there is a travel of mitochondria between cells and tissue, perhaps in normal conditions, this is healthy. And in lupus, for example, we see more mitochondria. Perhaps these mitochondria are diverse, they are oxidized or damaged, and there are antibodies against mitochondria, which may change the target of these mitochondria. Maybe they are then targeted to FC-expressing cells. Of course, those are hypotheses that are being developed, but I think this is where our field stands right now. So I wish to thank the collaborator, of course, Paul Fortin from Université Laval and the SLIC group, Christian Rood from University of Washington, all of the lab members.
Thank you, and thank you for keeping to time also, which gives us time for some questions. I'll just give some time for people to come to the microphone. Okay, no, they're leaving. Okay, sorry. So, yeah, you spoke very briefly about antiphospholipid syndrome, which I found intriguing, and something I don't know, just a thought that came to my head while you were speaking was that could this could these mechanisms have a role in obstetric features of APS, where we have both thrombotic and inflammatory features, but then in the absence of pregnancy, you get nothing?
That's a good question. And in terms of APS, yeah, I didn't develop much, but of course, it's another target of anti-mitochondrial antibodies. You can think of cardiolipin, for example. And although mitofusin doesn't seem to be a target in APS, but it needs to be replicated in more patients. It'd be interesting to see whether there's a connection with pregnancy, like you mentioned. That's clearly a topic that we find interesting, especially with the placenta, which may be shedding a lot of cells. It's known it's shedding DNA and perhaps mitochondria at the same time. So
that's vasculopathy, inflammation, but only in one context. There's another question over here.
Yes, thank you very much. Tadeucin from Ljubljana, Slovenia. Approximately 1/3 of patients with SLE developed antibodies against DNase 1L3. Did you check for this subtype of autoantibodies in your patients that developed anti-mitochondrial antibodies?
No, unfortunately, we have not. This is the big limitation to the study. We didn't have the luxury of looking at the other traditional antibodies. That's something that I would like to do, clearly, and more including the
DNase 1L3.
Yeah, that'd be very interesting. Just measuring these antibodies in these, we had 9,000 samples, that making the mitochondria and the RNA, that was our endeavor. And I think for the study of chronic diseases like that, we'll need chronic funding, which was missing. And but that's our goal, to actually monitor other antibodies and see if they if they replicate, or if there are a cluster of patients, for example.
Thank you.
Okay, and then just time for one more very briefly.
Hi, Caroline Jeffries from Cedars-Sinai. Did you look at a correlation between interferon score or the interferons with any of your mitochondrial antibodies?
Correlation with interferon?
Type 1 interferons.
No, same thing. No, we haven't measured that. I can tell you though, in mice we have, for what it's worth. But in mouse models, when we included the FC receptor on platelets, so that there's a release of mitochondria, which is not occurring in normal lupus mice, because the platelets in mice do not have a FC receptor for IgG. So we included that transgene. In these mice, we found that interferon was measurable in blood, which is not normally in mice with lupus, and mitochondria were present. That's the only correlation I can make in mice.
Yeah, I mean, your cardiovascular atherosclerotic association with the mitochondrial anti-RNA or anti-mitochondrial RNA antibody is fascinating, because the cardiovascular literature is really heading towards RIG-I, and RIG-I been important in cardiovascular disease. So, you know, there's something possibly there. So very nice work.
Yeah, it's a very good point. Yeah, thank you.
Thank you very much.
Thank you.
All right. Before I announce the next speaker, we haven't introduced ourselves. I don't think Professor Edward Vidal needs a lot of introduction, but he's a professor in translational medicine and rheumatologist in Leeds Hospital. And my name is Anna Burkic. I'm internist clinical immunologist and allergist at the Erasmus Medical Center in Rotterdam. And I would like to give a warm welcome to the next speaker. That's Dr. Rami Beshara. He's an assistant professor in immunology at the University of Paris-Saclay. And his work focuses on post-transcriptional regulation of immunity. And he will talk about the accumulation of mitochondrial RNA at the origin of autoimmune disease with anti-ribonucleoproteins. The floor is yours.
So thank you very much for this invitation. So like the other two speakers, I did not choose the title of my talk, and the typo you can see under my name, I'm not responsible about it. So, and I put origin into 2 brackets because I think it will be tough to really connect mitochondrial RNA to the origin, but I have a big duty today is to convince you that mitochondrial RNA is as important as mitochondrial DNA. in at least generating this interferon signature we see in these autoimmune diseases. So first, I will start by explaining how these mitochondrial RNA are generated, how they are sensed, and I will finish a little bit with what we are actually doing in the research in the lab of Professor Xavier Mariette. So I will take a step a little bit backward to remind you that RNA biology is much bigger than just focusing on mRNA. I'll use the mouse. So actually, mRNA account for just 1 to 5% of total cellular RNA inside the cell. And they took the light recently because of all the RNA vaccines and RNA-based therapeutics, which led 2 years ago to the Nobel Prize to Katalin Karikó and Drew Wisman. But the majority of the RNA inside the cell is actually composed of non-coding RNA, such as long non-coding RNA or tRNA, but also mitochondrial RNA. And these RNA can adopt several secondary structures. So I have a single-stranded RNA that's found in mRNA, but also upon certain conditions, these RNA can adopt secondary structure leading to this double-stranded RNA, which will be the focus of my talk. So historically, mainly or classically, this double-stranded RNA signature is mainly found during viral infection. So RNA viruses, upon replication, they can generate these double-stranded RNA. And within the evolution, our cells has kept a system of sensor that is able to recognize these double-stranded RNA. So you see them here, PKR, MDA5, RIGI, toll-like receptor 3. And the common point between all these sensors is that the outcome is really leading to the interferon type 1 signature among many other inflammatory inputs. But what is the origin of this double-stranded RNA? But before, I want to mention that all these double-stranded RNA are not sensed equally between the different sensors. So, for example, PKR sense basically double-stranded RNA that's in the cytoplasm, toll-like receptor 3 in the endosome, MDA5 double-stranded RNA that's long double-stranded RNA, and RIGI shorter double-stranded RNA, but ultimately we have the same signature, interferon type 1. And then what could be the endogenous source of these double-stranded RNA? So first major source is transposable element, which will need a lot of time to discuss, so I'm not going into it. We have RNA modification, the second cause. So like DNA, RNA can be modified, and there is some defect in RNA modification that could lead to a secondary structure in the RNA leading to this double-stranded RNA and then interferon type 1 response, but also mitochondria, of course. So mitochondria is our massive reservoir of RNA molecule, and I found this study published back in 2011 that quantified mitochondrial mRNA in comparison to other mRNA in the cell, and they found that at least 5 to 30% of the mRNA in the cells are composed by just 11 mitochondrial mRNA out of 13 mitochondrial mRNA. And this changed between the tissue. Of course, for example, in the heart, which is metabolically very active, so there is more needed about transcription of mitochondrial RNA, and the percentage even increase. And growing evidence recently is pinpointing to a link between mitochondrial double-stranded RNA and a lot of inflammatory and autoimmune diseases, which I will review some of them later in my slides. But I will start with one really nice example is Sjogren disease, which is an autoimmune disease that affects mainly exocrine gland, salivary gland, leading to dryness. And one of the first study to link mitochondrial double-strand RNA to Sjogren, it was published in 2022, where authors were able to quantify mitochondrial RNA in the saliva and tears of Sjogren patients. So you can see them here in red, more highly expressed, but also here. And some of these mitochondrial transcript actually correlated with the focus score. So you see here ND5, the heavy chain of NDA5 correlated really with the focus score. And these results were independently validated by another recent study showing that also in saliva of Sjogren patients, but also in their PBMCs, we are able to detect mitochondrial signature. And this, the score of the authors established a score that correlated with ISG or interferon signature in PBMCs and plasma, but also in some clinical score, as you can see here, SDI. So what I will really talk, it's 2 points today. Is first one, much easier. I try to convince you that really mitochondrial double-strand RNA contribute to this interferon type 1 signature. And the second one, I will speculate whether it can be really at the origin of these autoimmune diseases. But to answer these questions, we need to understand how mitochondrial transcription happens. So, in the mitochondria, the circular, as mentioned by Virginia, the DNA is circular, is very compact, but it can encode around 37 genes that are basically 13 protein encoding genes, 13 mitochondrial mRNA, 22 tRNA, and 2 ribosomal RNA. So transcription happens from both sides, the light chain and the heavy chain. And this will lead to the generation of 2 complementary strands of RNA and then increase the risk of generating double-stranded RNA. So during transcription, this RNA, mitochondrial RNA, is very long. And then through the action of several RNAs, it can be chopped to generate the mitochondrial mRNA, mitochondrial RNA, ribosomal RNA, and tRNA. And some proteins can bind to the mitochondrial mRNA and then stabilize this mitochondrial mRNA. And once we say stabilizing, it could also lead to an increase of risk of generating double-stranded RNA. And luckily, we also have some proteins that can degrade mitochondrial double-stranded RNA. And there's multiple proteins. I will cite 3 of them that I will talk about them later: SUF3, PNPT1, and REXO2. So first, we will start with the stabilization pathway with LIRP. And a recent paper published last year showed that actually in lupus and Sjögren patients in their monocytes, there is an increase in the expression of SLIRP. You can see it here. And in RA patient also here. And the mechanism proposed in this study that upon some stress that happens or take place through MDA5 signaling, SLIRP can move toward the mitochondria and then stabilize double-stranded RNA generated within the mitochondria, which then will leak outside the mitochondria, sensed by MDA5, and then induce a positive loop that leads to more interferon type 1 signature. But what about the degradation pathway? So luckily, as I told you, during homeostasis, the mitochondria is occupied by a system of degradosome protein that can bind to the double-strand RNA to degrade it. SUF3 will bind to the double-strand RNA and separate both strands. That will allow PNPT1 to bind to the single strand and start degrading the RNA. And then at the end, REXO2 can really generate smaller nucleotide. So one of the first studies that really assessed the link between these degradosome and interferon type 1 signature come from this paper in Nature, where authors were able to generate a technique to map what are the endogenous double-stranded RNA. So they took, I think, fibroblast, they they did cell lysis, and then they incubated the RNA with an antibody against double-stranded RNA, the J2 antibody. They IP'd the RNA, and then they sequenced it. And they found that the majority, a big portion of the double-stranded RNA that was immunoprecipitated comes actually from the mitochondria, because you can see here, this is the heavy and the light strand. And they found also that double-stranded RNA by this immunofluorescence co-localize with the mitochondria staining. And what happens when they knock down SUF3 or PNPT1? They increased, you can see them here, upon knockdown of SUF3, they increased double-stranded RNA generation, but also upon PNPT1 generation. And what I found interesting in this paper is that they found a cohort of patients that have mutation in PNPT1, and then PNPT1 cannot function properly, and then they showed that this was associated with an increase in double-stranded RNA generation in their fibroblast, but also an increase in type 1 interferon signature. Another recent paper was interested in checking SUF3, more specifically in Sjögren disease, in a cohort of pediatric Sjögren disease. And what they found in a subset of interferon-rich monocyte, there was a decrease in SUF3. So I remind you, SUF3 is one of the proteins that degrade mitochondrial double-stranded RNA. And this was associated with an increase in double-stranded RNA generation. And the working model is that if we have decrease in SUF3, then mitochondrial DNA could not be degraded, which then leads to more leakage of mitochondrial double-stranded RNA toward the cytoplasm, and then interferon type 1 signature. But in our lab, we were interested in studying this degradosome pathway and also in Sjögren disease. And the group of Professor Mariette, among many other here in the room, showed that epithelial cells in this disease are not only the target of autoimmunity, but actually play an important role in activating the immune response, whether B cell or T cells. So we wanted to check this degradosome pathway mainly in epithelial cells, real immune cell implicated in this disease. And upon checking the several degradosome, we found specifically a decrease in the expression of REXO2. So what I'm showing you here is immunofluorescence from a salivary gland biopsy of healthy control and Sjogren disease patient. In green, EPCAM staining for epithelial cells, and in red, it's REXO2. It's one of the degrader, mitochondrial DNA RNA degrader. We see that in Sjogren patient, they have decrease in REXO2 expression. And then if we knock down REXO2 in primary epithelial cells, we increase double-stranded RNA generation, which then we show that it's also part of it's colocalizing with the mitochondria and part outside the mitochondria. And this was associated with an increase in type 1 interferon, which also we check the sensor. So if you want to hear more about this story, go tomorrow to Alejandro's talk and you will hear more. But then we asked how could really, what could really drive REXO2 down regulation in Sjogren tissue. So we found that the transcript, the mRNA of REXO2 was heavily methylated. So like DNA, RNA can be modified. And there is a specific methylation called N6-methyladenosine, which we take epithelial cells, we immunoprecipitate them using an antibody against this modification, and then we quantify the enrichment, and we found a high enrichment in REXO2 mRNA in the IP. And this methylation takes place with the help of an enzyme, methyl-3, that will add the methyl group on the RNA. So we took advantage of the first pharmacological inhibitor of this enzyme called STM2457 to block the pathway and see what the outcome on REXO2. And what we found that we decreased REXO2 mRNA and protein levels. So our working model is that methyl-3 will catalyze the modification on REXO2 mRNA to increase its stability, then increase in its translation, which then will block double-stranded RNA and then interferon type 1 signature. But I didn't answer why it's downregulated in Sjogren patient. We published last year a paper showing that epithelial cells of controls, if we stimulate them with poly-IC or other stimulant, they can increase methyl-3 expression, which is an anti-inflammatory protein. However, cells from Sjogren patients lack this efficient upregulation of methyl-3, which we also confirmed by quantifying total M6A levels in the RNA. So we think that since Sjogren patients lack this efficient upregulation of methyl-3 in their epithelial cells, so there's no efficient methylation of REXO2, and then REXO2 is dysregulated and more type 1 interferon. The last part, we tried to reverse this phenomenon by checking a pathway that could increase REXO2 expression. So we focused on the methylation pathway. So methyl-3, once add the methyl group on the adenosine of the mRNA, it uses actually the methionine metabolism. So we had the idea of increasing or supplementing our media with S-adenosyl methionine. And what we found is that we increase actually REXO2 mRNA, we increase its half-life, its protein level, but also we downregulate at the same time type 1 interferon. I showed you one study showing that SUF3 was downregulated, and we are showing that actually another degrader called REXO2 being downregulated. So what makes the specificity? The answer is very simple. It's actually, if we check REXO2 expression across tissue, we found that's highly expressed in salivary glands. So we think that REXO2 pathway is really important in epithelial cells. However, SUF3 is you can find it here, and on the y-axis is really less expressed in salivary gland. So we think that really REXO2 is very specific for salivary gland. So I've showed you so far two important pathways, the stabilization pathway, the degradation pathway with these three proteins. But also let's not forget that the big role of mitochondria is generating cell respiration and these different metabolites. So actually a nice paper linked metabolite to mitochondrial double-stranded RNA generation through this fumarate metabolism or this fumarate hydratase enzyme. So what has been shown about this enzyme is that it's decreased in a lupus patient, I think in monocytes or macrophage. And upon knockdown of or inhibition of the fumarate of this enzyme, we have increase in type 1 interferon. What I found interesting in this paper is that they link it mainly to mitochondrial double-stranded RNA, because once they block the CGAS pathway, CGAS-STING pathways, they were not able to reverse the phenomenon. So it looked like the fumarate pathway could be directly or indirectly linked, but specifically to mitochondrial RNA versus DNA. How mitochondrial RNA can reach the cytosol. So far, nothing, I won't say nothing, but very few is known about how specifically mitochondrial RNA can leak in the mitochondria. What's suggested is mainly the same pathways as mitochondrial DNA, so BAX-PAC, VDAC, which is also a channel in the outer membrane of the mitochondria. And in our study, we found that in epithelial cells of Sjogren patient, once we block the BAX-PAC pathway, we were able to reverse this interferon signature. So I hope I convinced you that mitochondrial double-stranded RNA is as important as DNA in generating this danger signal leading to type 1 interferon. But so far I cannot answer why or how this model can really explain why RNA binding protein are targeted in this disease. So I will try to speculate with you. Maybe to answer this question, we need to remind ourselves that RNA are never naked inside the cells. They are always bound by proteins called RNA binding proteins. And it happens that classical autoantigen are RNA binding proteins. So took this figure from paper published last year that shows several autoimmune diseases has common or specific autoantigen, and most of them are RNA binding proteins. You can see them here in both colors, blue color. And another paper that was interested mainly in Rho60 RNA binding protein, and authors wanted to identify what could be the RNA bound by Rho60, so the immunoprecipitate Rho60, and then isolate the RNA, sequenced it. Of course, we know all that why RNA are bound by Rho60. What I found interesting is that there is a 10-fold enrichment in mitochondrial RNA, in some mitochondrial RNA. So it's just specific case of Rho60 showing you that it can also bind to mRNA. And this will get me to my working model that upon some stress, so mitochondria is stressed, we release mitochondrial double-stranded RNA, of course, DNA in some cases also, that are recognized by some sensors leading to type 1 interferon signature, which then will act in an autocrine manner and paracrine manner to activate immune cells. At the same time, mitochondrial RNA that are released from the mitochondria won't be naked in the cell. They are bound by some proteins. It could happen that they are autoantigens that upon apoptosis, for example, they could be released outside the cells, are recognized by autoantibodies, generate immune complex that could be also recognized by B cells, internalized, and liberate the RNA toward the endosome, activating toll-like receptor 7, 8, and then generating more interferon. It also could also be internalized in myeloid cells or PDC to generate also interferon signature. And with this, I think I would like to thank all the members from Professor Mariette's lab, and specifically the people who are bolded here. Alejandro, I don't know if he's in the room. Go to his talk tomorrow, ask him a lot of questions, you know more than me, and all the collaborators. And we are always looking for motivated people to join us. So there's multiple reasons why we have a nice view on the Eiffel Tower. We won the Champions League. I hope there's no Arsenal fans inside. And we are hosting the International Sjogren Symposium very soon. So registration is still open. Thank you very much.
Great talk, Rami. Really wonderful, because we're already 5 minutes behind schedule. There's short one.
Yeah, short one. So going back to my question before, you mentioned mitochondrial stress could sort of make start the chain reaction. So what is stressing the mitochondria then?
So for what we think, so in our research on Sjogren, we didn't find any problem in the membrane of the mitochondria. So we think it's going through, there is a defect in the degradation pathway, which then leads to more accumulation inside the mitochondria of double-stranded RNA, and then at some moment, these need to be leaked outside the mitochondria. So this is what.
Yeah, thank you.
Okay. Thank you. All right, then we move on to the last speaker of this session, and that's Dr. Serena Colla-Francesco. She's from Milan, University of Milan in Italy, and she's a rheumatologist and a scientist with a main focus on Sjögren's disease. And she will tell us today how metabolic remodeling of salivary gland epithelium links mitochondrial dysfunction to chronic inflammation in Sjögren's disease. The floor is yours.
Thank you for the introduction. So, as we know, Sjögren's disease is an autoimmune condition characterized by a chronic inflammatory loop developing within exocrine glands. And we know, as it has been said, that a crucial role in the pathogenesis of Sjögren is played by the local salivary gland epithelial cells. And some years ago, in this regard, we described how in epithelial cells from Sjögren, there seems to be evidence of a maladaptive activation of autophagy, which seems to not only to sustain their survival, but might be linked to their pro-inflammatory activity. But actually, the fact that we found such an activation of autophagy might suggest the presence of a metabolic rewiring. And that's the reason why we came up with the hypothesis that in Sjögren's disease, a bioenergetic adaptation of salivary gland epithelial cells can sustain and possibly drive the chronic inflammation. So in order to test this hypothesis, what we did first, we tried to find out whether actually there is a cause relationship between the inflammatory insult and the metabolic rewiring. To do so, we used in vivo model of ectopic lymphoid structure formation within salivary gland, which is a known mouse model of Sjögren. And by the use of transcriptomic, we looked at the metabolic rewiring occurring over the course of inflammation. And what we found was basically that if we look at the entire salivary gland, there is evidence of a metabolic rewiring which is induced by inflammation soon after the inflammatory insult and then recover over time. But if we look in detail to the salivary gland epithelial cells, there is actually evidence of a metabolic rewiring which is induced later on and is characterized by a downregulation of mitochondrial programs like OXPHOS and fatty acid metabolism. So after assessing this cause relationship, we then moved forward and we tried to assess and better characterize this metabolic rewiring occurring in epithelial cells from Sjögren. So to do so, once again, we started looking at the entire salivary gland. We compared Sjögren to Zika by transcriptomic analysis, and what we found was the evidence of a nice metabolic rewiring in Sjögren, in particular characterized by an overexpression of OXPHOS. But once again, if we look in the epithelial cells, in particular, this was done by a metabolomic analysis, what we found was a very profound metabolic rewiring. In particular, what we observed in Sjögren was an accumulation of different metabolites, and we were particularly intrigued by the accumulation of lactate, which might suggest an overexpression of glycolysis, but also the accumulation of different intermediates of the TCA cycle, which points at a potential problem in the mitochondria. So that's why we moved on and we performed some functional experiments by Sears analysis. And what we found surprisingly is that despite there is an evidence of an accumulation of lactate, there is no evidence in terms of functional activity of an overexpression of glycolysis, while there is actually a downregulation of the oxidative phosphorylation. So to better dissect the problem and find out where is the dysfunction, we also performed some functional experiments by using carbon tracing in order to see how the carbon is metabolized and passed from the glycolysis to the TCA cycle. And actually what we found by these experiments was not only a confirmation that glycolysis is not that overactivated, but in particular that the TCA cycle is slowed down, and also that the TCA cycle seems to be fed particularly by the glutamate, which is a mechanism that is known as anaplerotic TCA cycle that is very typical of cells having an oxidative phosphorylation dysfunction. So having found this dysfunction, we move forward and we try to find an answer on what is the cause of this dysfunction. So we performed lots of in vitro experiments, in particular using metabolic inhibitors like glycolysis and OXPHOS inhibitors. And what we found, despite a confirm of what I've shown so far, was interesting that if we inhibit glycolysis or OXPHOS in Sjögren, we can see how there is an accumulation of the NADH metabolites, which point to a problem in the redox mechanism mechanism, so of course to the mitochondrial programs. But also interesting, what we found is that even if we inhibit OXPHOS or glycolysis, epithelial cells from Sjögren are still able even better to produce ATP, which strongly supported the engagement of alternative energetic mechanisms. So since everything brought us to hypothesize a problem, of course, in the mitochondria of these cells, we used transcriptomic and proteomic to look inside this compartment. And here we found a confirmation how, if we look at protein level, there is evidence of a downregulation of different mitochondrial protein, and this is paralleled by an upregulation of gene transcription, in particular genes related to mitochondrial function. So finally, having found this mitochondrial dysfunction, we of course then ask the question, would it be possible that this dysfunction is linked and maybe drive their chronic inflammatory activation? So we performed lots of experiments in this regard. Here I'm showing only a part of them, but basically I'm showing these experiments which clearly demonstrate that if we treat in vitro epithelial cells with poly-SC, which activate their inflammatory activity, but then we dampen glycolysis, OXPHOS, or glutaminolysis, the major pathway that has an impact in their inflammatory activation is actually glycolysis. And that's the reason why we moved to the tissue and we try to find some evidence of a glycolytic impairment at tissue level. So we look at the expression of GLUT1, which is a major transporter of glucose, and we did not only found an expression of these markers on epithelial cells, but we also found how this was nicely correlated with an increasing severity of inflammation in the salivary gland. And also we found how the expression of GLUT1 is differential according to the different cell types, but more data for who is interested will be presented tomorrow in the forum session. So to conclude, in this study, we describe and we confirm how in epithelial cells from Sjögren, there is evidence of a mitochondrial dysfunction, which appear to be the cause of the metabolic rewiring that we observed at the beginning. This metabolic rewiring seems to be linked to their pro-inflammatory capacity. In particular, the major metabolic pathways which are involved are glycolysis and glutaminolysis. And finally, interesting, despite this dysfunction, we provide some evidence that epithelial cells from Sjögren are still capable in producing ATP, which does not only possibly suggest that this metabolic rewiring is just a consequence of inflammation, but that possibly this mechanism may have a role in the pathogenesis of Sjögren. So thank you for the attention.
Thank you, Serena, for the wonderful talk. Are there questions? One last question from the audience. Yes.
Hey, hi. How do the cells produce ATP without mitochondrial function?
That's a good question. So basically, we think that glycolysis is the major pathway. We know that glycolysis is less efficient because the amount of ATP is lower, but what we found in particular when we activate the cells, there was no time to show this data here, but at the basal level, they don't overactivate glycolysis, but once we stimulate them, they produce much more glycolysis compared to our controls. So we hypothesize that that could be one of the mechanisms, and also the glutaminolysis, which is another pathway where we concentrated to, we think that it could be another escape mechanism, in particular by the link with the TCA cycle that might contribute in the energy production. But of course, we have more data to answer your question.
Thanks.
Thank you very much. All right, then we close this session. I would like to thank all the speakers and thank you for you audience for the questions.